straightforward. So our sense of taste, this is our other chemical sense. And taste is actually 80% smell. If you have something wrong with your ability to smell, then you lose some of your ability to taste. Food tastes bland when smell is taken out of the picture. So the taste buds are the special sense organs for taste. Most of the taste buds are on your tongue in what we call papilla. What do they look like? Oh, they're bumps poking off of your tongue. What I tell you about papilla? They're bumps all the time. So most of them are on your tongue. You have some on your soft palate, which is at the back of your oral cavity, some on your cheeks, some on your pharynx, some on your epiglottis. What this can help with is when you're vomiting and tasting as it's coming out of your throat, it's because of all of this that's going on here. My stomach has been turning since 5 this morning. I'm burping and it tastes like fish. I haven't eaten fish in days. So there's something in my stomach and I'm tasting it because not of anything on my tongue, but because as I burp, all those chemicals are coming past my epiglottis, pharynx, and soft palate. It's wonderful. So just be aware of that. Most of your taste buds are on your tongue, but that doesn't mean that we can't taste from other places of our oral cavity. So each taste bud consists of 2 different types of epithelial cells. Our gustatory cells are the taste receptor cells. So they're called gustatory cells. Are the taste receptor cells. And they have these things called gustatory hairs that project into what we call taste pores and are bathed in saliva. And then they've also got supporting cells that get replaced every 7 to 10 days. So this is kind of showing you one of these things called a papilla, and then our taste bud, you can see, is this organ down here in it. So if we zoom into a taste bud, you see that we have 2 types of cells, our gustatory receptor cells, or our gustatory epithelial cells, and supporting cells. The gustatory cells have these hairs on top, and they're all surrounding this taste pore on your tongue. So your tastins suspended in saliva are going to run past this taste pore and get picked up by those gustatory hairs. So we've got 5 basic taste sensations. There's starting to be a growing body of evidence which suggests that we have a 6th taste sensation. So sweet, sour, salty, bitter, umami. Umami is the flavor that you get, that savory flavor in like meat and tomatoes. And again, they're starting to think that maybe we have lipid taste buds, which is why I love McDonald's French fries so much. So, so to be able to taste a chemical, it must first be dissolved in saliva, and then it has to diffuse into the taste pore. And then it's got to contact our gustatory hairs. And each of the different sensation is going to cause different ion channels to open, which is why it gives you different sensations. And it's way too much detail to go over right now. So you can take that. I think I took that in neurophysiology as a master's degree student. So from our taste bud then, where do we go? From the taste bud, our impulses from the anterior 2/3 of our tongue are going to come along our facial nerve. From the posterior 1/3 of our tongue is going to carry the impulses from the posterior 1/3 along our glossopharyngeal nerve, which is not number 10. Ooh, that's a misprint in your book. Number 10 is the vagus nerve, and that's what's going to be responding to like your fish burps in your epiglottis and pharynx. So if we look at that then, we would stimulate taste buds here in the tongue or here in the tongue. Our first order neuron is either the facial nerve, the glossopharyngeal nerve, or the vagus nerve. So we would go from our taste bud, or we could think from our gustatory cell to our first order neuron, which is either facial, glossopharyngeal, or vagus. And that's actually going to synapse in your brainstem. So the information from that first order neuron is going to synapse in your medulla oblongata, and then our second order neuron is going to ascend to the thalamus. So this would be we have little receptive endings here, and then we'd be coming along, our soma's here, and we're synapsing onto our second order neuron in the brainstem. Specifically the medulla oblongata, if you want to know. And then from there, we're going to ascend to our sensory relay station, which is the thalamus. And from the thalamus, we're going to synapse where? Where's the gustatory cortex? So here's our second order neuron is going from the brainstem to the thalamus, and our third order neuron is going to go to the No, this is the weird one, the insula. So the insula contains our primary gustatory cortex, and that's where we would have our conscious perception of taste. Taste is important also because it helps trigger reflexes that do things like help you salivate or increase gastric juice into the stomach. So may initiate protective things like gagging and vomiting. So other things to be aware of, taste is 80% smell. Your mouth also contains thermoreceptors, mechanoreceptors, nociceptors. I mean, we were kind of aware of that. So some things might be enhanced or detracted by taste and by texture and temperature. Some people excite pain receptors in their mouth with things like spicy foods. And some people hate that. They feel it as pain, but then some people love it. They feel it as pleasure. Pleasure and pain activate the same types of pathways in your brain. It's fascinating. So, yeah, that's that for our chemosenses. Any questions? All right, then let's quick run through the anatomy, and then we'll look at physiology tomorrow. And you'll have the rest of today to look at models, and then tomorrow we'll do a cow eye dissection. So, for the anatomy of the ear, we break our ear. I don't have enough models to give everybody a model, so I just brought one for me. We look into three major areas of the ear: our external ear, middle ear, and internal ear. So, if we look here, the external ear has two parts, what we call the auricle and the external acoustic meatus. So the auricle, or the pinna, is this part here that has this cartilaginous rim called the helix and a fleshy lobule, an earlobe. So those are the two parts of the auricle. And the auricle, the shape of it is such that as sound waves are going through the air, it funnels those sound waves into the next part of your external ear called the external acoustic meatus. So we'll write that down. As far as the function of the auricle, it's to funnel sound waves into the external acoustic meatus. So, the auricle funnels sound waves into the external acoustic meatus. What kind of glands do I find in my external acoustic meatus? Ceruminous glands. So yeah, that's just, bring it all together. The external acoustic meatus, then, the auditory canal, is going to bring those waves into what people call the eardrum. It's actually called the tympanic membrane, and it's the border between the external and middle ear. So we'll say that. Our external acoustic meatus brings the sound. Whoa, whoa! What is even going on? Where is that thing?
It's underneath the model.
Sorry. Craziness. Okay. So So our external acoustic meatus is going to bring our sound waves to the tympanic membrane. And this is what people call the eardrum. It's the border between the external and middle ear. And it vibrates in response to the sound waves. So it vibrates when sound waves hit it. So we can see structure of the ear, external ear here, this is the auricle, the cartilage rim is the helix, the lobules down here, leading into our external acoustic meatus, which is bringing sound waves to the tympanic membrane, which is going to vibrate. And what's it going to vibrate? These bones in our middle ear. So, is everybody good on this? Shout no if no. Okay, so your middle ear, also sometimes called the tympanic cavity. Now, really the only thing that I care that you know about in the middle ear are your auditory ossicles. They're the smallest bones in the body, and they're going to, as that tympanic membrane vibrates, it's going to rock the auditory ossicles, and they're going to then beat into this place called the oval window. The oval window is right here, and it leads into the structures of our inner ear. The cochlea is the organ of hearing. And so now we're going to vibrate this, which rocks these bones, which knocks on the oval window, which is going to create waves in this fluid that's in here. So we'll look at that in just a minute. But as far as what I want you to know for the middle ear are the auditory ossicles. They're called the malleus, the incus, and the stapes. So you can see the malleus is here attached to the tympanic membrane, which is then attached to this one here called the incus. The incus is attached to the stapes, and the stapes is knocking on the oval window. So, like on this model right here, what I can see is I can pull out my tympanic membrane, and my malleus and incus are on it. When I pull out my cochlea, there's the stapes. So you can't actually see the oval window, I don't think, on any of our models, because our stapes is attached to it every time. So then just be aware of that. So this stapes is going to vibrate, and now we're going to come in here, there's a membranous labyrinth full of fluid, and we're going to convert that to a wave in fluid now that will excite our organ of hearing. So any questions? Do I need to write anything down from that? You'll see those names again. The middle ear contains our auditory ossicles, the malleus, the incus, and the stapes. Do you want me to write something down?
Well, no. So those are the three smallest bones, but when you won't be able to see the stapes in the model because it'll always be connected.
Because they leave it attached to the oval window.
Okay.
Yeah, so we have some real ossicles that I'll pull out for you to look at. They're super cool. And maybe I shouldn't admit it, but one semester we may have dissected the ossicles out of a cat's head. It was awesome. I'm definitely clipping that out of my YouTube video because it's probably not ethical. Well, I guess it's not unethical per se, but it wasn't in the lab. We just did it. Okay, so that's all I care about for the inner ear. The middle ear. Then we're going to go into the inner ear. This is also called the labyrinth or the maze. And so it's in your temporal bone, right behind your eye socket, and we divide it into what we call the bony labyrinth. And these are just channels that go around inside there and is divided into these three spots. And inside the bony labyrinth, we find the membranous labyrinth. So the vestibule and semicircular canals are part of this bony labyrinth that are involved with equilibrium and head position. So I'm not going to test you on the details of them, okay? So this is involved in equilibrium and head position, and we're not going to worry about that special sense. We're going to worry about the other ones. The cochlea is the windy part of it, and this is what's involved in audition. So we are going to talk about that. So audition is your ability to hear, and we will talk about that. So inside the bony labyrinth, what I'm looking at right here is bony labyrinth. All of this that I'm holding. Here are my semicircular canals. So I think your lab has you identify them. So sure, be able to identify that those are semicircular canals. I'm not going to ask you about the physiology of them. This part right here leading into it is the vestibule. Be aware of that, but I'm not going to ask you about the physiology. This spirally part here is the cochlea. We are going to worry about that because that's what's involved in audition. So inside this bony labyrinth is a membranous labyrinth. There are membranous sacs that are full of fluid, and that's what's going to, when our stapes knocks on that oval window, we're going to vibrate that fluid, and that's what's going to excite our organ of hearing. So, as far as like anatomy goes, you would be expected to just be able to identify, okay, those are the semicircular canals, this is the vestibule, here's my cochlea. And so this is showing you the bony labyrinth pulled back and the membranous labyrinth in it. And the cochlea is your organ of hearing, and it wraps around and around in spirals. We call the organ of hearing is called the organ of Corti, and so that's found within this membranous labyrinth, and we'll go through that probably more tomorrow, just because we don't have any models that are going to do anything like this. But as it winds around and around, we've got, it forms what we call the spiral organ, or the organ of Corti, and we'll look tomorrow at how we excite that. So those are our structures. Any questions on the anatomy of the ear? Okay, so anatomy of the eye. We have accessory structures, and those are involved with the eyeball. We'll talk about our accessory structures tomorrow because I do want to talk about the functions of them. I just wanted to go through the anatomy of the eye right now, so when you're looking at your lab, you know what you're looking at. So for our eye, it's got 3 layers. Our eyeball is a pretty complex organ, and you can see it's also got all these extrinsic eye muscles. I'm not going to make you worry about the extrinsic eye muscles. I'm going to make you worry about the layers of the eye. So we've got 3 layers. A fibrous layer, outer layer, that's what we're looking at. On the outside of all of your models, that's the fibrous layer. And it's got 2 parts. The cornea, this clear window in the front, and the sclera, the white of your eye. So here is the fibrous layer. Cornea here. This is just a clear window. It does not do anything with light, but let it in. It protects the interior surface of your eye. So that's part of the fibrous layer. And then the sclera is the white. So that's our fibrous layer, the outermost layer. Intermediate, we have the vascular layer. So back on this picture, here's part of it. All this stuff in the back is called choroid. Choroid means black. This is going to help catch light waves and stop them from dispersing. So it helps catch them and focus them on the photoreceptor cells that are important. So for our vascular layer, our second layer, it's the pigmented middle layer, the pigmented layer of the eye, and that's one part you need to know about, the choroid. But then you also need to know about this part in the front, the ciliary body. So the ciliary body has these little muscles and ligaments that are attached to your lens. Your lens is the first structure that does something with light. It refracts it and focuses it onto your retina. So, and the way it can do that, we can accommodate and change the shape of the lens for whether we're looking at something up close or something far away. So this ciliary body is what's going to do that. It's got these muscles that are going to pull on these ligaments to change the shape of your lens for close or distant vision. The other part of the vascular layer is your iris. So that's what we see right here. Your pupil, I did not know that the pupil was a nothing until I dissected a cow eye in 201. Your pupil is a nothing. It's a hole in your iris. So your iris, my iris is blue because it doesn't have melanin. Everybody's irises have different melanocytes in it that give them different colors. And so the iris is this part that can either dilate to allow in more light or constrict to allow in less light. And so your pupil is just the hole that's changing size. And the reason it looks so black is because black absorbs all light. So as light is coming in, we are absorbing it all in our inner layer. The retina is what's absorbing it all, but it's making it look black because there's nothing to reflect black back at you. So I think those are the only important parts that we need to worry about for our vascular layer. So this is showing you our iris is controlled. It can be constricted when we've got parasympathetic going. It's dilated when sympathetic's going. We'll talk more about that tomorrow. The inner layer is called the retina, and the inner layer has two parts again. It's got your retina has two layers. This pigmented layer on the back, which is very thin, and then this neural layer right here. So the pigmented layer is going to help with the choroid to trap light and keep it concentrated so that it doesn't disperse. Because if I have black here and pigment here, then when the light hits it, I can trap it there. And now these, which are my photoreceptors, can respond to it. Where right here, where the light's hitting. As opposed to if I didn't trap it, it would scatter and then I would have inefficient processing. So for our retina, again, it's got two layers. The pigmented layer and then the neural layer, which has our rods and cones. So the only other thing that we'd be aware of anatomically is that we can split it, the eyeball, into anterior and posterior segments. The lens and the ciliary zonule separate the two. So this is going to be what's cool when you dissect your eyeballs, is that the posterior segment back here is full of this fluid called vitreous humor. Vitreous humor is a fluid, but it's so thick that if you're careful, you can hold it in your hand as a glob. So it's a fluid that is like almost solid because it's so thick. So if you're not careful, it's going to just